presentation. So my name is Ralf. I'm from the University of Freiburg, and the reason why we actually focused on carotenoids in carrots came because I'm in the lab of the co-inventor of golden rice, Peter Bayer, and we are interested in basic research to improve the storage, the amount and stability of carotenoids, and that's why we came to carrots, because carrots is actually a prime example of high amounts of carotenoids which can be stored for a long time. And I think I can go through this quite quickly because Florent already showed this scheme. But anyhow, I would like to focus on phytoin synthase, which is the first enzyme actually of carotenoid biosynthesis, and actually removes the carbon from the common isoprenoid precursors, as here GGPP, which flows also in chlorophyll, tocopherols, and so on. Phytoin is colorless, and after desaturations and isomerizations, is converted into red-colored lycopene, followed by cyclizations, which yields alpha-carotene and beta-carotene. And these carotenes are hydroxylated by a total of at least 4 hydroxylases, at least in Arabidopsis, and yield the different xanthophylls. And last but not least, special or specific xanthophylls are cleaved into abscisic acid, a plant hormone. It's been known since many years, actually, that phytoin synthase has a key role, which already Florent told. And again, the tomato examples is the most famous examples, or the most impressive example, because phytoin synthase expression is induced during fruit ripening of tomato and correlates very nicely with the formation of lycopene, the main pigment of tomatoes. Another example is deethylation of seedlings, for example, Arabidopsis seedlings. Phytoin synthase is induced during deethylation and correlates with the enhanced synthesis of beta-carotene, alpha-carotene, and xanthophylls to build up the photosynthetic apparatus. And we found also that, especially in poaceae, rice, maize, but also in cassava roots, phytoin synthase, or I should say specialized forms of phytoin synthase, are induced during abiotic stress situation and correlate with the enhanced synthesis of abscisic acid. So all these indicate phytoin synthase is really the rate-limiting enzyme of the pathway and determines the amounts of carotenoids which are formed. So we asked what happens if you overexpress phytoin synthase, and I should say this is done in many tissues, and in most tissues increases the carotenoid content also. We did this in Arabidopsis, and actually exclusively in non-green tissue, we have seen a large increase in carotenoid accumulation. On the left side, you see callus tissue, which we induce from a germinating seedling on auxin-containing medium in the dark. And you clearly see that they accumulate large amounts of carotenoids. These carotenoids are mainly beta-carotene, small amounts of alpha-carotene, really small amounts, some intermediates which are non-colored, so phytoene and phytofluorine, which is gray in the drawing, and small increases in xanthophylls. Here you see the phytoin synthase protein levels, so the overexpressed transgene. On the right side, you see the same analysis done with the roots of these transgenic lines. So a similar picture, you see large increases in beta-carotene, mainly phytoene, phytofluorine. So we find increases of about 10-fold or 100-fold even for roots. So normally, if a tissue accumulates carotenoids, it produces accordingly structures to capture these or to sequester these carotenoids. They develop chromoplasts, but in Arabidopsis it's not known to contain or to develop chromoplasts. So we wonder how Arabidopsis stores these carotenoids. And we were surprised to see that these carotenoids are simply stored as crystals. within the cells. So this means, as an explanation, if you simply increase the biosynthesis by increasing the phytosynthetase activity amounts, beta-carotene crystals sequester automatically. So you don't need a developmental program, which is normally used or believed to be necessary to develop chromoplasts in tomatoes, for example, but also for carrots. So we thought, and also with the pattern of mainly beta-carotene, phytoin, phytofluorine, and the crystal formation, whether in carrots the same principle might be involved, so also phytoin synthase. And to test this, also this is a picture which we have seen right now. We overexpress the bacterial phytoin synthase in a white carrot background, which results in a strong accumulation of carotenoids, which are also sequestered as crystals. This is the top right picture, which you see. On the top, on the bottom right side, you see, for comparison, a picture of an orange cultivar, Chantenay red cord. So we see overexpression of phytoin synthase leads to enhanced carotenoid accumulation in carrots, and also to the formation of crystals. So we moved on and checked whether phytoin synthase is also involved in the accumulation of, correlates also with the high accumulation of carotenoids in orange cultivars, and found that in 2 orange cultivars which we checked, this was Chantenay red cord and Nantes, the protein amounts of phytoin synthase correlate nicely with a high accumulation of carotenoids, while in 2 white colored varieties, Queen Anne's Lace and Küttinger, we almost could not detect any phytoin synthase protein. Again, the question with if these protein levels correlate with the expression level of the phytoin synthase genes, it's hard to answer. You see some slight differences normally if you compare orange with white carrot cultivars, but it seems that these huge differences in protein amounts that you see require some additional translational or post-translational mechanism. So, however, if you compare the pattern of carotenoids which accumulate in transgenic psi overexpressing white carrots in the middle and orange cultivars on the right side, you see that there is a major difference, and this is the additional accumulation of alpha-carotene. It's shown in red here. We did not see alpha-carotene accumulating in psi overexpressing white carrots, and we also did not see alpha-carotene in Arabidopsis, which overexpress phytoin synthase. So this means that alpha-carotene is something specific for orange cultivars. We went back to Arabidopsis because there is a mutant, it's called LUT5, which is deficient in a hydroxylase at this position. This deficiency results in an impaired hydroxylation of alpha-carotene. So what happens in the leaves, so we're looking now at the leaves. In leaves you accumulate large amounts of alpha-carotene in addition to beta-carotene. Normally in the wild type leaf of Arabidopsis, you see only weak or very, very low amounts of alpha-carotene. And interestingly, we found that leaves of orange carrots similarly accumulate higher amount of alpha-carotene compared to white-colored or white-rooted variety. So we thought that maybe this high abundance of alpha-carotene is also linked with a deficient alpha-carotene hydroxylation activity. And to test this, we overexpressed the LUT5 gene, so the CYP97A3, under control of a root-specific promoter in orange carrots. And in fact, this is what happened. So the alpha-carotene content was indeed reduced. Skip to the next. You see better. So on the right side is the pattern of these transgenic lines, which contain only 2% alpha-carotene. On the left side, the control, the non-transformed control, so they're shown in a red cord. So we reduced the alpha-carotene content to about only 10%. This is just to show the expression of the transgene, and the gene is also, so the protein is produced. However, so this is what we have expected. However, what was quite unexpected was that we reduced the total amount of carotenoids, which you can see, because actually we would have expected that the amount of alpha-carotene which is converted into xanthophylls accumulates. So actually we would have expected that the amount of xanthophylls increases, which is not the case. Moreover, we have seen a decrease in beta-carotene in the transgenic lines, and also of phytoene and phytofluene. And this normally indicates a reduced activity of phytoene synthase. So what we first checked was whether these transgenic lines showed a reduced expression of phytoene synthase. But as you can see, both genes, Psy1 and Psy2, show a similar expression level. So we could not explain the reduced carotenoid content with reduced expression of the genes. However, we found a nice correlation between the protein levels of phytoene synthase, which were reduced by about 50% in these transgenic lines, which then nicely correlated with the about 30 to 50% reduction in total carotenoid content. So how can this be explained? And this is the model which we suggested. We suggest that there is a constant negative feedback, which is derived from xanthophylls acting negatively on phytoene synthase protein amounts. And when we produce higher amounts of these xanthophylls or the derivatives thereof, these negative feedback inhibition of phytoene synthase protein amounts is even stronger, and this would explain the reduced protein levels we have seen in the transgenics. So all these data indicate that the high abundance of alpha-carotene in orange carrots is really due to alpha-carotene hydroxylation deficiency. So we wondered whether we see some mutations, some alterations in the carrot LUT5 gene. The LUT5 from carrots was the only one which was not sequenced. So we sequenced it first from a white variety, Queen Anne's lace. It shows 71% identity to the Arabidopsis gene, is about 600 amino acids long. We find a single copy gene in white and orange cultivars. However, the expression of the LUT5 gene was similar in the white and orange colored carrots. So we could not explain the phenotype we have seen by just simply reduced expression of the LUT5 gene in the orange carrots. However, we found an insertion in the LUT5 gene of orange colored carrots. It's an 8 nucleotide insertion, which leads to a shorter protein, which is missing a critical cysteine residue, which is required for the binding of the cytochrome P450. So this enzyme has to be considered as non-functional, and actually explains the phenotype we have seen. So to generalize these findings, we made an association mapping analysis with the insertion deletion, which we have identified for the LUT5, and find a nice correlation with the alpha-carotene amounts and alpha-carotene to beta-carotene ratio in a carrot population, which we have investigated. So I come to my conclusions. I've shown you that phytoene synthase is considered as the rate-limiting enzyme in many cases. The overexpression of phytin synthase results in the accumulation of carotenoid as crystals, and this happens in both Arabidopsis, non-green tissues, as well as in white carrots. Phytin synthase also correlates with protein levels, with carotenoid levels, comparing orange with white cultivars. However, the high alpha-carotene abundance in orange carrots cannot be explained by simply high phytin synthase levels, but it is similar to the Arabidopsis LUT5 mutant. The root-specific overexpression of the LUT5 gene from Arabidopsis results in reduced alpha-carotene content, but also in a reduction of total carotenoid amounts, which correlates with lower Psi protein levels and suggests a negative feedback. We found a deficient LUT5 allele in orange carrots, which encodes a non-functional enzyme, and found a nice association of this polymorphism with high alpha-carotene levels. Time to say thank you to the University of Freiburg, Peter Bayer, for this support. Jacobo Arango is now moved to CIAT in Colombia, who did the carrot transformation and a lot of clonings, and Carmen Schubert for his technical skills. And here in Angers, Emmanuel for the association studies, and Mathieu Jourdan also. And also Jérémy Cloteau, who is not here, but who is the one who made the link between Freiburg and Angers. And those who financed the work, Harvest Plus, and grant from the Deutsche Forschungsgesellschaft. Thank you very much for your attention.
Thanks for the presentation. Is lutein in yellow carrots stored in a crystalline form?
It's a good question. I don't know. Probably not. Probably not. Because we think that this the formation of crystals prevents the carotenoids from being degraded, and also depends probably on the structure. And to see the structure of lutein, I would rather say it cannot, and it's maybe probably more prone to be degraded than beta carotene.